Okay, in this video, we're going to look at DNA translation. This is simply the process that is needed to synthesize proteins or create proteins. And I'm going to break it down in a very different approach. We're going to take the kitchen approach. So we have a recipe if you want to cook up something, right? What we want to produce are proteins by the time translation is done. So mRNA, which is messenger RNA, ribonucleic acid, is going to be the set of instructions that is needed for this process to happen. So that's like our recipe. Then think about ribosomes to be the place where the whole synthesis is taking place. It's more like our what? Our kitchen. And the ingredients are simply going to be the amino acids. So these ingredients are going to be brought to the ribosome by a special helper which is called tRNA, the transfer RNA. So brings this to the ribosomes. So tRNA does the transferring for the whole process, the cooking to start. And we're going to ensure that the amino acids are added in a matching order according to a specific code or a certain protein that is called for. So we're going to be following the recipe. The ribosome reads the mRNA in groups of 3 letters. That's what we call a codon. So a codon is simply a combination of 3 amino acids. Yeah, so I'll get into the definition of what a gene is, genetic code. I'm just trying to give you an overview of the topic. So a codon is simply a 3 sequence of amino acids, a 3 base sequence. So I can give you something like this. Let's say AAG, something that looks like this. And now each codon tells the ribosome which amino acid to add next. The tRNA brings the correct amino acids and the ribosome is going to link them together. So tRNA just brings a particular sequence, 3-coded base paired sequence of this guy here, amino acids. Then the ribosome will be arranging them in a particular order. So that is going to be explained using the genetic code. Let me just give you a picture of what we're going to be looking at. So this here is what we mean by the sequence, the codon, right? The codons that are going to be coded. Yeah, so if you combine, let's say U, C, and A, so you have UCA, meaning you are going to produce, you can either you are producing this guy here, that's what you're having, that's the amino acid that is being coded for, right? So you can have leucine, meaning you combined U and let's say G, right? That's what you have, depending this side. So you have this letter, let's say the first letter is U, then the second letter is U, then the third letter is, let's say, G, meaning you've produced leucine using these 3-coded amino acids. That's basically what the transfer RNA is going to be bringing, and the order in which the amino acids will be arranged. So let's say you have leucine, then the next guy is arginine, for instance, meaning you had this guy followed by this other guy here. So the ribosome is the one packing them depending on what type of protein we want to create or synthesize. Yeah, so that explains the first part. Now we said a codon is simply a sequence of 3 bases. That's what we call a codon. Now we have to follow the recipe, right? That's what we said. And building up the protein, as the ribosome reads the mRNA, it builds the protein chain by linking these amino acids together. This chain will eventually add up until you produce a functional protein for a particular function. So every protein is needed somewhere in the body, right? And when the ribosome reaches a stop codon on the mRNA, there are certain stop codons that are going to be encoded for, because we have to put a stop to these things. If we don't, the amino acids are continuously going to be added. The ribosome will continuously add the amino acids codons that are being brought by the tRNA. So we need to put a stop to it whenever a satisfied or a desired protein has been synthesized already. The protein chain is going to be released so that it can fold into its original shape. You remember how proteins are formed. You can have a primary, tertiary, or quaternary structure ready for it to do its job. So it depends on the type of protein that you need. So basically, in a nutshell, this is what we're going to be doing. Okay, so we're just saying DNA translation is the protein synthesis, which is simply a major second Okay, so let's dive into the full detailed process of how DNA translation takes place. So a recap on the actual definition of DNA translation. It's called the protein synthesis, the production of proteins, and we can also say it's assembling of amino acids into polypeptides based on the genetic information carried by mRNA, right? From DNAs, that's what we have. And it's a very complicated process. The good part is that it's precise because it's a second major step when it comes to gene expression. Yeah, so it's a very big step. Yeah, so it's precise. Yeah, because it's a second major step from transcription. Replication is not considered to be a big step because replication, like I said earlier on, it only happens because DNA is a double helix type of structure. So we have to split it open. But the actual translation of the DNA blueprint information into something that can be useful starts under replication. Yeah, so this is what we have. So it involves the participation of all the types of RNAs. So we're going to have messenger RNA, tRNA, and RNA in the ribosome, of course. And we have a number of enzymes that are going to be needed or proteins that are going to be produced. And the length of these polypeptides is going to be dependent on the genetic information carried by the messenger RNA. Yeah, so there should be a stoppage. Like I said here earlier on to say there's going to be a stopping time, a timeline sort of. There's a termination to the whole translation process. So in cells, mostly in eukaryotic cells, it's going to happen in the cytoplasm. So we're going to have the whole mature mRNA being transferred from the nucleus and going to the cytoplasm where the ribosomes are located for the whole translation to occur. But mostly, we're just saying either prokaryotic or eukaryotic cells, we're going to have the whole process happening in ribosomes. Now, these ribosomes are going to attach to your messenger RNA, of course, and start being transcribed, the whole translation process. And it's going to be done in a 5' end of messenger RNA, whilst the 3' is the end. So we're going to start with the 5' to 3'. This is the direction that the translation occurs. Now, there are a few things that you have to learn in terms of the terms. I'm sure you've done this in class. Things like a genetic code, which is just like a DNA template, so to say. So messenger RNA is a linear molecule, which varies in length depending on the type of polypeptide that you are coding for. Yeah, so it's read according to the genetic code, which is a sequence. It's a linear sequence. And a codon, like we said, it's a 3 base type of arrangement, type of a sequence. Yeah, so that's what we call a codon. And sometimes we call it a triplet code. So we have that particular expression we can use. So a combination of these codons or triplet codons is what is going to produce a chain of amino acids that we end up calling proteins because they're going to be polypeptides. So when we talk about the genetic code, it's just a sequence of nucleotides on the messenger RNA derived from DNA. Remember the kitchen recipe thing I was giving us? So it's a set of instructions in a gene that tell the cell how to make a specific protein. That's basically what we have. Yeah, so this set of instructions, a genetic code is simply a set of instructions in a gene that tell the cell how to make a particular specific protein. It's like a universal triple nucleotide. It's like a rule that we're always going to be having 3 bases. So it's going to control the whole protein synthesis using a triplet code of bases. Yeah, so whenever we have our 20 amino acids, we can have a codon which have 3 bases on either DNA or messenger RNA, which can now be translated or be transferred into a set of instructions that we call a gene. So a gene is simply a sequence of bases on DNA which code for a sequence of amino acids in a polypeptide. So that's what we define. So notice the difference between a gene and a codon. Yeah, so a codon is a sequence of 3 bases, either on DNA, so sequence. So this is a sequence of 3 bases. Then this guy here, a gene, is a sequence of bases on DNA which code for a sequence of amino acids. Yeah, so I'll give us an example in terms of explaining the major difference here. So let's talk about the codon first, under the genetic code. Yeah, so we have codons, right? So let's say genetic code. Yeah, so the first thing that we have is we have codons, which are in linear triplets. These are linear. So we have, let me just put the name. So we have codons. Then 2, we have non-overlapping. So we have non-overlapping. So during this whole translation process, codons do not overlap. So codons are like pairs, right? Like they always say, let's say you have this. Let's say, imagine this was possible, right? For us to have such a case. Yeah, then this is your G here. Then you have A, U, G. Yeah, so these guys here are the codons, these triplets that you're seeing. Now, these are not overlapping because they're not joined. Yeah, so the codons do not overlap but read sequentially, 3 at a time. So we read them 3 at a time. We can just say they are commaless. All nucleotides are used for code for amino acids, with none of them used for punctuation. So they're just going to be read in a, like there is no comma. I was not supposed to put any demarcation between these guys. There is no separation. There's nothing separating them because initially we believe it's one thing connected. Yeah, that's what we have. So these are universal because they're going to be found in all organisms. Because of their polarity in terms of them having a constant or permanent reading, which is 5' to 3' direction. We can say they are degenerate. That's one of the characteristics as well. So we have one codon, one amino acid can be formed from different encoded triplets. So like leucine, you see how the combination of CUU, CUC can still give us leucine, right? This is what we mean by they are degenerate. Yeah, they are all calling for the same type of amino acid. And the ones that are going to be the nonsense codons or the stop codons are the ones that we'll be using for. So these are very important. You need to master these. They like asking about them. We call them stopping codons. These are used to bring about a stoppage to the addition of these triplets in the ribosome so that if a complete protein has been synthesized, we need to terminate the whole elongation process of your polypeptide. We need to introduce one of these. These are going to be the nonsense, or rather they are the stop codons. Yeah, that's what we have. Now, to understand this, we need to talk about what we call the Wobble hypothesis. Remember, don't lose track here. We're trying to explain what happens when a protein is needed, or rather when we are creating a protein. Remember, you're coming from a background where you know what proteins are. You know what type of food stuff sometimes contain proteins and everything else. Now, we want to talk about those proteins that are synthesized in animals or in the bodies of living organisms. That's what we want to do. Yeah. These are important proteins that are needed for continuity of life, and also to get certain features transferred from one generation to the next. That's what we just need. Yeah, so when we talk about the Wobble hypothesis, let me give you a proper illustration here. This is going to lead to the genetic code, but we have to understand it first. So let's talk about the Wobble hypothesis. So I just didn't want to read the notes today. I wanted to try to use this approach in biology as well so that people can follow the same way they follow the other courses as well. So firstly, the Wobble hypothesis explains how our cells can read the whole genetic code efficiently. It shows how the transfer RNA molecule can recognize more than one type of codon, which is a sequence of 3 nucleotides, right? That's what we call a codon to be. Yeah, on the mRNA. So tRNA needs to specifically read a certain type of sequence on your messenger RNA. Remember, the messenger RNA is the one that we're going to be using to carry the message in terms of giving the whole expression in the ribosome. So when this happens, there are 64 possible outcomes that can happen, because we're having a combination. We're having four nucleotides. The sequence on a codon, it has three amino acids. So the 64 type of combinations or the possible outcomes is a result of, you have in RNA, you have possible 4 bases, right? You have adenine, you have guanine, then you have uracil, and you have what? You have thymine, right? Is it? I know you have cytosine. Yeah, you have cytosine. So we're saying these 4 guys, we need 3 of them, right? So meaning that's where the 64 is coming from. These are the 64 possible ways that we can pick or have a combination of 3 amino acids from the 4, these guys here. So these are the possible outcomes that we can have. And we only have 20 transfer RNA amino acids that we can bring to the ribosome. So how do these molecules recognize the different types, or rather the possible codons that we need? This is where the Wabo solution comes in, this hypothesis. So it proposes that since we're going to be reading the anticodon on the tRNA, that pairs up with the codon, of course, in a 5 to 3, 5' to 3' direction. It's not a rigid type of process. So the Wabo, which simply means like flexible, it allows different pairs of nucleotides on the 3rd position of the codon. So how it works is like this. If I give you a proper example, so let's say you have a single tRNA that you are given, which can of course recognize multiple type of codons calling for the same amino acids. So we have UC. Imagine you have UUC, and you have U. These guys are both going to call for phenylalanine. That's what they call for. So if we go on our table, if I show you here on our table, we said we have UUU, you see this, and also UUC. They are all calling for the same type of what? Amino acid. So what happens is that since they can both call for the same type of amino acid, so we're having U, and you're having UUC. The tRNA with the anticodon, let's say A, G, which is red in there, because this is happening on the mRNA. Remember, this is like your DNA, right? This is what you're having. This is the sequence of the amino acid being brought in. So we're creating a template which is going to now be read in the opposite direction, which is 3' to 5'. The AA is coming as a result of uracil. Uracil is related to what? To A, right? Then you have uracil again to A. You have cytosine, which can be to G, or uracil, which can again be A. Yeah, so we're saying the AAG can pair with both these guys here. Instead of limiting the choice of attracting, or rather pairing up the cytosine to guanine only, we're saying since they're both producing phenylalanine, we can use this template to put an end, or rather to put a full stop to what we have. So this creates a Wabo where we have a choice, which is just a flexible choice between combining UU triple U to AAG, in as much as uracil is not supposed to be linked to guanine at the far end. But because there's another codon, which is UUC, which codes for the same amino acid, phenylalanine, we can also use the guy that stops this. So it's also, you can think about it like this. Let me just ask us a simple question to see if people are following. Look at this. What is the codon on the mRNA that is going to be coded for when we have UUU? This is going to be AAA, right? So not only can 3' and 5' this sequence be coded for this or match with this guy, it can also be paired with this, because it's also, this UUC was also producing the same one, the same type of amino acid. Yeah, so it explains the efficiency in translation to say cells don't need a separate transfer RNA for every codon making proteins. Whenever we are trying to synthesize the protons, we don't need a particular type of codon. So this makes this whole process a little bit efficient and also flexible, because the genetic code is going to be flexible, allowing for some variation in codon recognition. That's basically what we are saying. So it's like a clever trick that we can say that helps our cells to read the genetic code efficiently, which in turn gives us a variety in terms of, you see how people can be born from the same parents, but they look totally different from each other, right? So that offers a little bit of variety in the streamline to the whole process of protein synthesis. Yeah, so I hope that explains the Wobble process. This is the genetic code that I was using to just explain what we mean by the Wobble process. And now we're supposed to look at the different types of RNA that we can study at your level. So you know how you have your messenger RNA and you have your transfer RNA, and then from here you have the ribosomal RNA. So just let's talk about their functions. I know we did this under transcription, but we'll talk about it a little bit again here. So mRNA, which is our messenger RNA, which is synthesized from transcription. Remember, this comes from DNA. This just carries the information. So I'll just give us 5 functions on why we need messenger RNA in translation. So it carries the genetic information. That's number 1. This is from the nucleus to the ribosome in the cytoplasm. Then second, so the first one carries the information. So it's a messenger, right? That's what we have. Then it offers some form of specificity when it comes to the amino acid sequence. Yeah, because it's the one that codes for proteins. It's the one that brings, it's a messenger bringing the message as to which protein is needed. So it's going to ascertain the order of, or rather the sequence of the amino acids. So it's the one that determines the sequence. Then the third rule is that it provides some form of template. Yeah, because it brings a strand, right? One strand, so that the synthesis can happen on using transfer RNA for a complementary template to be formed. Yeah, so it's the one that provides a template. So we can have this. Then the fourth one, it initiates the whole translation process. So the start codon, which is either, we just say initiation, let's just say initiation. It brings about the triplet codon, AUG, on mRNA, which signals the beginning of translation and directs the movement of the ribosome. So it determines the movement of the ribosome, reading the mRNA and assembling the amino acids accordingly. Yeah, that's basically what we have. So it directs ribosomes. Yeah, so we can have this. Then when it comes to transfer RNA, yeah, so before we talk about the next guy, remember we said AUG is the initiation codon, then UAA, UAG, or UGA. These are termination or rather stop codons. You have to know this. They like asking them when it comes to multiple choice questions. Okay, so here is a quick overview of the roles of tRNA. So it acts as an intermediate. It's transferring, right, from messenger RNA to the polypeptide that we need, the proteins that we need. So it's a small molecule which is about going to contain about 80 nucleotides. These are the base pairs that you're going to have, the triplets that you're going to see. Each amino acid has its own transfer RNA. So depending on the type of protein that is being synthesized, so every triplet codon has its own transfer RNA. It's just a temporal form of DNA, right? That's what we have. So this is going to be carrying what you call an anticodon. So right here, we're going to have there's always a sequence which runs from 5' to 3', which is used to attach to that particular amino acid. So this is at the end of each transfer RNA, there's going to be this sequence, this CCA, which is going to be used more like a stopping type of sequence. Not really stopping, but signifying that we are done with the first triplet. We need to add another triplet. Yeah, so it's an adapter molecules connecting, right? That's what we have. They have one end that can read the triplet code in messenger RNA through the complementary base pairing. Another end which attaches to the incoming or either. So if you are having something that is coming like this, already there's already a sequence of triplets that were already in portion. So if this is what is being brought in place, so we're saying at the beginning, so this is the beginning of this guy, then this is like the end of this sequence to this side. So the joining, complementing this other side, that's what your transfer RNA is going to do. And we have this type of tRNA structure that you can master. The only thing they'll ever ask is to label these structures. So we have things that we call like the D arm. It's always going to be like this. We have the D arm, the variable arm, the T arm, the amino acid acceptor arm. These are the elongated part which contains the hydroxyl. Then we have a phosphate, this guy, read in as 5', then the hydroxyl as 3', because we're going like this. We're rotating this thing like that. Yeah, then we have the anticodon down here. So let me just show you the important thing. So before we even do the next part where we talk about the whole process in terms of the mechanism of translation, like the binding of the messenger RNA to the ribosome, the elongation, everything else, let's talk about the last type of RNA, which is ribosomal, which makes up of 80% of the total RNA in a cell. It combines with proteins to form ribosomes, which are the site of protein synthesis. So not only do we make these proteins in the kitchen, but we need to make some of the proteins for the kitchen, which is some of the proteins are going to actually be formed into ribosomes. Yeah, and these are very important. So it has two subunits. We have what we call small and large, S and large, to hold the mRNA and tRNA associated with enzymes in position until a particular peptide bond has been formed between subsequent, rather adjacent amino acids. So we have what we call an amino acid site, which is the A site, and also a P site, a peptidosite. So each subunit is going to exist separately in the cytoplasm, but two can join together on the RNA molecule. So let me show you this using your lecture notes, something that is a little bit more illustrative. So we have what we call the peptide site and the amino site. So this is what you have. When you have the 30 small ribosome, the initiation complex, combining with the 50S ribosome 50S, these are always going to give you 70S. That's the math that we do. 30 plus 50, we get a 70. So we're going to have the two subunits combining to give us this type of expression, which has a peptidosite, this side, and an amino acid site. That's basically what we get whenever you combine ribosomes, the 30S, the 30 subunits and the 50 subunits. So the whole process is going to happen in a following manner. We have this mechanism that summarizes the whole translation mechanism. So we have the binding of messenger RNA to the ribosome. So this happens already from the ribosome in the cytoplasm. So it's going to have a small ribosome unit which binds to the messenger RNA near the 5' cap. So there's going to be, we have some form of initiator transfer RNA, which is going to be carrying the initiating codon, which is AUG, like we mentioned previously. And this is recognized because once this is exposed in the ribosome, the whole process starts. It positions the alignment of the fellow triplets, the fellow codons that are going to be coming after it. Yeah, so we're going to have a continuous binding of the ribosomal units on all the amino acids that are going to be produced. We're having these templates being produced, so we're going to have some form of amino acid activation. That's basically what we have. The P site, the two sites that we talked about, the peptide site and the elongation is going to be happening. So in the elongation, let me just try to put this here. Yeah, so we have the most important thing is the addition of this. This is the 30S and the 50S, which we talked about. And the next thing that we can have is, okay, the activation of amino acids. Yeah, so, okay, no, this is irrelevant. So things that are irrelevant that they'll never ask you about are what I'm skipping. I'll just focus on the part which is necessary. Okay, so during elongation, this continuous process, the ribosome moves along the messenger RNA, reading the codons, the sequences of the 3 nucleotides, and adding the corresponding amino acids to the growing polypeptide chain. So the process is going to be involving the following steps that I'll just list here. There is, okay, this whole thing here, let me just summarize it here. There is what we call codon recognition. I'll just call it CR, codon recognition. The ribosome reads the messenger RNA codon by codon to recognize if we have encoded the right thing. Then there's going to be transfer RNA binding. So molecules carrying specific amino acids are going to bind to the ribosome, the transfer anticodon pairs with the messenger RNA, ensuring the correct amino acid is being added. So that's the tRNA binding. Then the third step, so this is tRNA binding. Then the third step is the peptide bond formation. So this is going to be a continuous elongated type of polypeptide bond that we're going to have. So the whole process is just going to be elongated until we reach a P site, so that we have termination, which occurs when we have those 3 types of combination of bases aligned whenever you have the stop codons or the nonsense codons. So under the termination process, you're going to have what we call release factors that bind the ribosome in response to the stop codon to stop it from adding more nucleotides to the sequence. Or we have the polypeptide release, which is a completed polypeptide chain, which is released from the transfer RNA in the P site. Or the last part, we can just talk about the ribosome dissociation. It disintegrates to form back the small subunits that it had, until, because now the functional protein has been produced, a sufficient polypeptide chain has been achieved. So the subunits on the ribosome needs to disintegrate or dissociate to reform back the 30S and the 50S that you had, so that it gives chance for the functional protein to fall into a 3D dimension structure, sort of. Yeah, so however, they try to give you this, like, this is just for your own information. If they try to explain this in all the notes and the amino site, all this stuff that they're saying here. In eukaryote, just the differentiating here, this is, you have your ribosome doing its thing there with methionine, the amino acid that attaches to the ribosome to keep it active. It's like an initiator. Yeah, that's what you have. And you have a stop codon, you have the initiation, the whole complex. I'm only interested in the termination part because all these are going to be the same thing done over and over again. Yeah, so I want to talk about this part here. So this should be a correct way to represent what we've been talking about, the movement from 5' to 3', and every ribosome that you're going to have is receiving these guys that you're having. Yeah, these type of triplets, codons that you're going to be having. So it's going to be matching or putting the corresponding pairs depending on what type of amino acid we want to call for. So you're seeing these are the names. So when we pair up these are the names that we're forming. It has to be a precise process because it converts all the genetic code carried by messenger RNA into a particular sequence. Yeah, forming these cellular functional proteins. It is basically the fundamental process to the the whole central dogma thing, because it ensures continuity of life. When this has been misdone or done wrongfully, it can cause serious problems in the whole preservation of genetics. Yeah, so it enables these guys to synthesize the proteins that are needed correctly. So hope this is going to shed a little bit of some light as you are doing your personal studies. Yeah, I was just focused on things that you will need. Of course, there is a post-translation type of thing that we can talk about, but it's really irrelevant. Yeah, these all things here, they'll never ask you about them. This is just for your own information, really. Yeah. So if you do have any questions, you are studying anything, you have any particular question, please do reach out. For those that have registered in bio, thank you so much for watching.